I'll wait just another minute or two to let a few people more sign in. Thank you, everybody, for showing up this afternoon. I hope this is the beginning of a successful series of mini seminars and meetings. I know there are many things on people's minds, and so this is not the only thing you have to do with your days.
It's so quiet.
I apologize. This is the second time now this has happened. I'm not sure why Zoom doesn't like this particular meeting. I use Zoom reliably all day, every day, but for some reason this meeting seems to have an issue. So let me try that again. I'd like to welcome everyone. Can people see this? Before I start, can people see the screen share?
Yes.
Okay. And if I drop out again, Reinhardt is a co-host, so he can take over the meeting. And I apologize. I don't know what happened just now. So I'd like to welcome everybody again to their first IMAG MSM working group regular meeting and mini seminar. I'm pleased to see that you're here. I appreciate it. We're going to have some brief administrative items followed by a mini seminar by Yaling Li, who's been very generous to lead off these series of mini seminars. And then we'll have a discussion about organization of the working group for our remaining time. So just to remind you, I'm James Glazier at Indiana University, co-lead with Reinhard Laubenbacher, who's on Zoom. We thank Jim Sluka for the website of things, and Bruce Shapiro, who's been coordinating and handling emails. First thing I wanted to say was that we've been a little overwhelmed with the membership enrollment. We've had 125 people sign up from a large variety of countries. We did a little bit of demographics. About 71% of our members are from the US. About 93% are computer scientists, 20% call them experimentalists, and 4% clinicians. 62% academic. And then there are also a number of people who are industrial or regulatory or funding agency related. Jim, did you want to say a word about this dendrogram?
Yeah, so in the registration form, everybody answered, I believe there are 22 questions about your interests. You're a modeler interested in ODEs, or you're a biologist interested in innate immune response. Well, this is a cluster analysis of those 20, well, a subset of those 22 questions. This particular analysis just took into account the biological questions. So whether you're an ODE modeler or a spatial modeler doesn't contribute to this graph. So this is a graph of basically biological interest constricted by those questions that we actually had in the application form. One thing that jumps out is the entire top half of this graph. All these people with the larger dots picked either innate or adaptive immune response. So this entire top half of the graph is people interested in the immune response. The immune response, both innate and adaptive, the two groups overlap very, very heavily. About 90% of the people that are in one of those groups is in the other. I shouldn't call them groups, they're selections in the registration form. We're working on analyzing some of these other clusters. There's a cluster down at the bottom that appears to be people largely interested in outreach and educational aspects. As I said, the top graph is very heavily into the immune system, and we're still trying to sort out what some of these other groupings suggest. But we hope to be able to use this to some extent to help define what working groups we should have, what subgroups we should have in the working group. And that's all I have.
Okay, thank you. Okay, we have a Slack set up. We'll reshare the Slack invite in the chat for you. We have 11 channels to find so far. We hope that we can have additional channels to find as the subgroups for the organization of the working group come in. We also hope that the various leads for the various subgroups will be willing to monitor the Slack channels. At the moment, we're very thankful to Giuliano Gianlupi, who's been doing the Slack management for us. We've had a reasonable number of posts so far. Hope people will use it. There was a very nice suggestion that there should be a weekly topic of interest or questions of interest to discuss. This week's is about zoonotic origins of pathogens. And so anyone who would like to weigh in on the issues of zoonotic origins of pathogens is most welcome. We'd love to have your suggestions for good weekly questions or topics to highlight in the Slack. And so please get back to us with your ideas on that. So I don't want to take up too much of the time. Our next agenda item is our inaugural mini seminar by Yaling Li of Lehigh University. And I will turn over the screen share to you for your talk.
All right, thank you very much, James. Let me share my screen. All right. Are you able to see my screen?
Yeah, but you are.
Let me see what it's.
I think you're sharing in swap presenter view and slide view. Shell would do it.
How about now? It works now?
That's great.
All right, great. Thank you very much. All right, great. It's my honor to start this mini seminar series. So today I'm going to start with a very smaller scale, talk about some of my recent research on study the binding of SARS-CoV-2 to cell membrane, where we use a multiscale modeling approach on trying to link molecular information to the whole virus and then try to search for some potential drug inhibitors. So I will talk a little bit about background about ACE-RBD inhibition strategies, and then we use structure-based screening to search for potential drugs, and then we also try to use multiscale model to link molecular information to the whole virus scale. So first I need to say that I'm not really an expert on this topic at all, because my original research is mostly study nanomedicine design and fabrication and testing. I do both coarse-grained and diffusive particle dynamics modeling of how to design different coating of, such as antibody coating for different types of nanoparticles, and how does this kind of coating will enhance the adhesion and help nanoparticle to penetrate through the cell membrane. So we do both modeling as well as some experimental work. where we test and flash those nanoparticles and then quantify their binding to inflammation-induced cell membrane. But since the pandemic, I find there's a lot of similarity between nanoparticle and virus, because if you look about the general like entering pathway of virus, you will see that the virus also have a surface coating, which is those spike protein, and those spike protein will bind with ACE overexpressed on the cell membrane. That will reduce their entry energy and then ramp up on the surface of those membrane, and then you will see the enzyme TPRS2 will cleave the spike protein and eventually activate the virus. So the virus will break the membrane and then enter the cell and then bind into cytosis and then reproduce self. So I feel there is a lot of similarity between nanoparticles and the virus in terms of the entry process. So so this is definitely a multiscale problem in the sense that you will see that the whole virus has size about 100 nanometer. Well, if you look about the binding site, which is the spike protein, it is in a scale about tens of nanometer. And if you look about the real binding interaction, which is usually this RBD protein over here, it's in a scale about a few nanometer over here. So that's why we have to use different strategy to model those different scales. Use full atomistic simulation for the RBD protein itself, and then use coarse-grained molecular dynamics for the virus spike protein. And for the whole virus, we have to use even coarser scale modeling over here. So this is definitely a multi-scale host-pathogen interaction problem. So there's different strategies people usually use to inhibition of RBD and ACE2 binding. So for example, if you look about one smart design people proposed recently is because RBD would bind the ACE2, if you can design some peptide that mimic the structure and binding functionality of ACE2, then your RBD would preferably bind with your peptide first. So the peptide will occupy this binding site, then your RBD cannot bind with the real ACE expressed on the cell membrane anymore. So this is one of the strategies people use. So obviously there is more general like search for drug, which is actually you can repurpose a lot of those FDA approved drugs or small chemical compound to target the ACE2 and then this RBD interface over here. So you have quite a few targets, say you can target either RBD or ACE2 or the complex of those two. And then we can usually use so-called structure-based screening to search for drugs and then use molecular docking to find which of those drugs will actually bind best with your target protein. And there is a huge library of FDA-approved drugs or chemical database. You can actually search for a different type of drug and then score or evaluate whether they bind well or they bind bad. So this is the typical searching process. So usually we use the tool, what is called the molecular docking process. So basically you're going to search for a different drug molecule and then different poses of those drug molecules because they're able to rotate and then change their configuration. And then you can actually use different genetic search algorithms to figure out what is the best pose that will fit with those druggable pocket on your target protein. So in general, this kind of docking algorithm will generate like structure that very agrees pretty well with the crystal structure people observe in the physiological condition. So what we do over here is that we want to search for some potential drug that will inhibit the binding of ACE2 and this RBD site over here. So we're looking for some druggable pocket, such as this type of pocket that is showing on the, let's say this type of pocket in the middle, right, where you have interface, that's where we care most about, or another drug pocket that is RBD site over here. That's another potential druggable pocket. And then we want to find whether there's a drug that can bind firmly with those pocket on those interface. So the general process is pretty simple. You basically pull out those protein crystal structure from protein data bank, as well as library of drug formula. And then you add hydrogen bond, minimize your structure. And then you use different force field or potential. and try to find druggable pocket using molecular docking to find which one is energetically favorable for drug pose. And later on, you can also perform different types of molecular simulation to confirm dynamically it will perform inhibition effect. So the first thing we start with is this popular drug, HCQ. You probably heard a lot about that in the news, so that's where we start. What we find is that it has not very high binding affinity with ACE2, which is this red, which is this green pocket over here. And the docking score we got is about -4 kilocalvin per mole, which is kind of in the low side over here. But we took it and then we perform a steer molecular dynamic simulation to analyze the effect of HCQ on this complex binding. What we do is we pull the RBD from the ACE binding site at a certain rate, and then we attach the RBD with a virtual spring, and then so that we can measure like when you slowly pull it and how large the pulling force is required to separate them. So the reason we want to measure this kind of separation force, because people find from literature, this pulling force is linearly proportional or correlated with binding free energy between two proteins over here. So which means that once you figure out this maximum pulling or separation force, you pretty much get a free binding energy between those two proteins. So this is one of the tests we did. We put the complex together, and then we have a case where we add drug, and another case we don't add the drug. So the drug is docked over here. And then we measure during this separation pulling process, what is the change of force over time. So you will see that the black curve is without drug, and the red one with HCQ docking at this side over here. So we do observe that the force will increase first and then drop, right, which is the separation process. And then with HCQ over here, you will find it that your pulling force does decrease by about 20% or so. So which kind of indicate that this HCQ does reduce your pulling force or inhibit this type of binding. But the problem is that it doesn't bind very firmly, as I said before. The docking score is only about -4. So which means that it doesn't bind that firmly, but once it binds, it seems to be inhibit this binding separation. So we keep moving on, trying to find some drug candidate that will both bind firmly as well as inhibit this kind of binding, or make the metastable complex destable over here. So we target some docking or pocket that is at the interface between the RBD and ACE. So you do see that there is a kind of a pocket that is empty that might be a target point for the drug to fill in over here. So we search out two drugs that we feel that looks promising. One is this compound called SSAA09E2. It is actually a drug compound people screened out in 7 years ago for the first round of COVID, not COVID-19, but the previous round of SARS-CoV virus. And then people report this kind of drug might potentially interference with the binding of those two. And another promising candidate we find is a cancer drug, FDA-approved cancer drug called Nilotinib over here. It shows a promising binding score from the recent literature report, but they didn't actually further check whether the binding of this drug will do inhibition or do nothing. So, but they report that it has potentially a good binding score over here. So we take from here, and then we first study the binding of this SSA09 with both RBD and ACE2 as well as the complex. So what you can find over here is that the binding score of either RBD or the AC is not very high, -5, which is kind of reasonable. But the binding score at the complex pocket site is really high, which is -8. So which kind of indicate that this drug will likely bind with the complex rather than individual of those two. So it's kind of an interesting finding over here. And the same happens to actually for this cancer drug, Nilotinib over here. It has pretty high binding score, -8.32 on this docking site at the complex over here, which is very similar to the SSA, but they kind of bind as two upper side. One is at the front side, another one at the back end over here, both at a similar interface, but different side of this pocket. So the next thing we study is we study the root mean square dynamics of both drugs. So this one is for the ligand itself, which means that we want to see whether this drug will bind firmly. So you will see this SSAA bind pretty firmly. So the root mean square displacement of this drug kind of very stable. It doesn't fluctuate. Well, the other one, the nilotinib, has a lot more fluctuation, which kind of means that their docking is not very, very stable. It's kind of bind and then off, on and off. So it's not as strong as SAA. So if you check about the root mean square displacement of the complex, RBD ACE2 complex, we observe similar trend. So the black one as well, the red one is the baseline. You will see that this is without any drug, which is the red curve over here. It kind of locally fluctuate. But once you add in this drug SAA, you will find it that the root mean square displacement will have much larger fluctuation, which kind of indicate that the overall complex structure is not stable. It's kind of fluctuate up and down or on and off frequently. And if you look about the particular residue that is triggering to this fluctuation, we can find it that majority of those fluctuation of the residue is at the beginning part, which is from sequence from 300 to about 400, which is actually located at the interaction between the ACE2 and RBD over here, which kind of indicate it's the contact interface between those two proteins is fluctuating a lot and become unstable under this drug SSAA over here. So nilotinib also have a little bit fluctuation, but not as much as SSAA protein over here. So if you look about the molecular dynamic simulation that tells more detail, you will find it that the SSAA has very good docking. It's kind of bind very firmly over there and then make the overall structure fluctuate and unstable. While this nilotinib over here, it's kind of partial stable, right? You will see that it fluctuate and part of it is anchored over there, but it has another end that is dangling around. So which explain why it doesn't actually bind that firmly or doesn't actually inhibit that well over here. So from this one, we find it that this SAA might be a promising drug that both bind firmly and also make this complex structure unstable once it's binded over there. So of course, in order to confirm this, you also want to check about what's going to happen at the whole virus scale, because what we study or majority of the drug screening is only focused on a small portion, right? One single pair of RBD and ACE2. But as you realize that the cell membrane, we might have multiple of those ACE protein that bind with multiple of those spike proteins over here as well. So that's why we're also interested to see how does this kind of RBD ACE binding will influence the overall uptake or entry of this whole virus over here, which is the virus uptake process. So what we do over here is that we use a coarse-grained approach, because obviously with millions or more than millions of atoms, it's impossible to study use atomistic simulation. So what we do is that we coarse-grain, grouping different groups of atoms together into those beads. So we can actually simplify each of those RBD and ACE with hundreds of or thousands of beads rather than millions of atoms over here. So we use what is called shape-based mapping to map all atomic structure into those coarse-grained beads over here. But the key thing is that we need to develop a proper coarse-grained force field. And the requirement over here is that we want to preserve the flexibility or the dominant mode of those moving spike proteins, because recently there is quite a few people reporting that the spike protein itself is not rigid. It is actually swirling around or rotating around, and there is an average angle. So it's a very flexible structure. Such flexible structure will also help the spike protein or the virus to actively capture or encounter with those ACE protein in an active way. So we actually want to preserve the feature of this flexibility over here. And we also want to preserve the interaction between those proteins as well as the mechanical properties of the whole virus. So, so this is still ongoing work. We're still developing the potential. But if we're able to figure out a proper potential, we can develop this kind of whole virus scale coarse-grained model, right? We have all those distribution of those spike protein and then preserve its mechanical properties as well as its binding with ACE side over here. So for this binding part, again, we want to actually preserve the binding free energy. If we use a very simple Nino-Chunus potential, then we need to tune the potential parameters so that the overall binding energy will be equal with the binding energy obtained from all atomistic simulation over here. So we're trying to preserve the binding energy both internally as well as between RBD and AC protein over here. So once you obtain that kind of binding energy or coarse-grained model, then you can actually run steer molecular simulation or umbrella sampling to sample how large the force is needed to push the virus into the host cell surface, or how large the uptake force or uptake energy is supposed to be. So you can actually plot out the free ramping energy as a function of engulfment depth, which means that as a deeper and deeper the cell engulf into the cell membrane, we can actually plot out the change of the total energy. So if we say the binding energy or binding energy surface is called A, you will find that when the binding energy is very small, it's hard to uptake or hard to enter, right? The total energy will decrease and increase back. But if you have higher and higher binding energy between RBD and ACE2, you will find it that it becomes much easier and easier to uptake, which means that it's more favorable for the virus to enter the whole membrane when you have stronger and stronger or more and more those binding sites over there. So of course, another thing is that how did you know your model is correct, or how did you validate the model? So I'm working with one of my colleagues at Lehigh University, Frank Zhang, who was working on FM measurement of this RBD binding. So what they do is that they can conjugate RBD on an FM tip, and then they can coat a layer of AC receptor on a cover glass, and then they will put this FM tip close to the surface and then pull it. So you will see that they're able to measure the unbinding or the pulling force at different like pulling rate over here. And then you can also check out when you have blocking or inhibiting drug over here, how does this kind of blocking or inhibiting drug will reduce this kind of a separation force. So you can actually use those type of molecular scale validation data to validate the model if you want to verify your model is correct. So for the last two slides, James asked me to share about some potential direction that we can collaborate, as well as whether there is some interscale connection. So one collaboration opportunity I'm looking for is trying to use data-driven approach to develop some coarse-grained force field between ACE2 and RBD. As I told you, we haven't really figured out an accurate or good way to derive this type of coarse-grained potential. As you can imagine, you have tons of atom group you need to group together, and each of those group you actually need to derive their coarse-grained potential over here. So that's why in the literature, people actually use machine learning data-driven approach, because we have a lot of data from all-atomistic scale already. And how do we feed those data properly to derive those potential in a data-driven approach, so that we can actually derive in a fast and accurate and preserve our flexibility for this pair of potential, both for the coarse-grained approach as well as for the whole virus scale. I hope that some people who's familiar with machine learning or data-driven or potential development could collaborate on this type of topic. So another one is for connection between different scales. So obviously, my work is mostly focused on molecular scale, interaction between spike protein AC2 to the whole virus. But I believe that there is a lot of room at larger scale. For example, the AC expression organ scale might be heterogeneous, right? Depends on different location or different organ. You might have totally different AC expression density, and then that will also trigger different uptake process. And then maybe we can also couple with some larger scale mathematical infection models. I believe this is some work from James' group, where they studied the spreading of those virus, but coupling with this uptake process might help the spreading modeling as part of the input over there. So other topic could be that we can also study other type of protein interaction, or the fusion of the virus, or the overall ramping process. So I'm looking forward for discussion and collaboration with people in this working group. Thank you very much.
Thank you very much. That was great. Thank you for leading off. Appreciate it. We have time for just a couple of questions, if there are any from the audience. Jacob.
So you're trying to connect, you're working at the virus level or like molecule level, eventually to the cell level. This is the levels, and you're trying to reach higher up to, let's say, you eventually want to do a spreading of disease, so probably up to the population level. What ideas you have to put in the middle? What other ideas you have to reach up to that level?
That's a good question. Frankly, my immediate answer is no, I don't know how to do it. I feel this is definitely not a concurrent model, right? This is more like a hierarchical model. You derive from molecular scale, get this binding affinity, and then you will got, I would say, maybe the probability of entry and uptake, and then that will link to maybe the density of virus at local point. And then maybe you can link it to the distribution of ACE2, and that will actually couple to the organ scale. So I believe it's more like a hierarchical coupling rather than concurrent coupling, more serving as input of the next level.
Okay, I'll try to rephrase my question. Maybe I'll get a clearer answer, because it's hard for me at that level that you're talking. Please help me. So
okay.
I'm trying to see if I can get a characteristics, let's say, of an individual, because you have those nice molecules and you can compute some sort of probability that it will attach or not attach to the cell. So far, I'm correct?
Yeah, that's right. It's a molecular scale or a single virus.
Good. So if you have a single virus, and let's say you can compute from a single virus its ability to attach to the cell. Let's say you have other properties of a human being. Let's say height, weight, number of cells, I don't know, something, or number of susceptible cells. Can you give a probability of the virus attaching to that human and causing the disease? Is it something that is like foreseeable, or what things do you need to actually compute that?
That's a pretty complex question, I would say. First of all, you will see that in order for the first thing you need to say is how many virus will really enter, right, or contact with the cell.
Okay.
And the question I'm addressing is that after the contact with the cell, what is the chance or what is the probability it will enter the cell? After the enter cell, then the next thing is how many of them will produce themselves and then spread out. So there's multiple steps. I think what I'm addressing over here is one of the intermediate steps. Is if a virus is contact over here, what is the chance it's going to enter?
Good. So you already have some of the intermediates. If we can figure out some of the other ones, we can rise up to the population level. And the question is, who's in this group is able to do those other intermediate steps?
I think there are people in the group who are interested in barrier function. There are people in the group who are interested in airway transport. There are people in the group looking at ciliary transport and mucus. And so I think there will be, as time goes on, we'll see people filling in many of these pieces. And I think it's great that you bring up some of the issues that would have to be addressed if you wanted to have a whole sneeze to infection model. There's a lot that would be required. Is there time? We have time for one other very short question. I'm afraid that we have to stay on a tight schedule to keep it a mini seminar. So if there's one more question, you're welcome.
So yeah, if I can ask a quick question, I'm actually interested in particle that could be viral particle transport in the airways. But so to bind to these ACE2 receptors, do we have in the group expertise in what's the distribution? Where are those ACE2 receptors located in the lung? Are they mainly in the airways? Are they mainly in the gas exchange region? Because that's going to affect the distribution. The particle deposit non-uniformly in the lung. So that's an important aspect also.
Well, certainly different cell types have very different levels of expression of ACE2. So, and even within a single cell type, I'm sure there's a significant difference.
Yeah, so that's probably something that will need to be looked at.
Yeah, that's definitely some misinformation because we do actually search in literature about this critical information as you just asked, and we didn't actually find much clear data on that, in particular quantitative data. There's only like some description, like some people find like from the tissue sample where there is ACE2 overexpressed, but there's no quantitative data about the distribution among different like locations.
So that would be a good point to raise in the discussion groups, that that's missing information,
Yeah.
critical missing information. Okay, any other quick, I hope people can continue this discussion offline through the Slack and elsewhere. Thank you again, Yaling. That was wonderful first talk.
All right, thanks, James.
If you could, I'm going to go back to our less interesting business talk now. And let me just come back to that. Does everybody see the business slide deck now again? Okay. So thank you. So I wanted to come, first thing I wanted to talk about was that we are going to have these every week, we hope. We're going to try to do 2 a week, 1 or 2 a week. Try to keep them short. Next week, we have TJ Segal from Lehigh University, Gary Ann from University of Vermont, have very generously agreed to talk. The following week, Alan Perlson has very generously agreed to talk about his in-host viral modeling. We have one more slot for the 19th, if somebody would like to speak. We do not have speakers yet. The next week is Thanksgiving. I assume people don't want to meet on Thanksgiving Day. If you would like to meet on Thanksgiving Day, you're most welcome to ask us to. December 3rd, 10th, and 17th are available. Jacob has volunteered to speak one of these days. So please do let us know if you would like to speak, if you have people in your group who would like to speak, or suggest speakers to us. And now I'd like to come back to the business side of the meeting. Reinhard, you're welcome to speak up. I'm happy to turn this over to you in a few minutes. We'd like to go over the subgroup topics and provisional lead volunteers. People are very generous in volunteering to lead subgroups. We'd like to try to, by the next week, have leads for all the subgroups we're going to continue, consolidate subgroups without leads, because there's some which could be related but don't have lead suggestions. Split subgroups if they're needed. Ideally, in terms of principles of organization, self-organization, we'd like people not to lead more than one subgroup because it takes enough energy to lead one. But if somebody needs to lead 2, then that's okay. Some people very generously said they were interested in a lot of different subgroups. And so if you could give us a ranked preference, if you put yourself down for 6 possible subgroups, indicating the ones you care about the most would be very helpful. We will share the working document in the chat, the link, so people can edit that themselves. We'd like to have 1 or 2 co-leads per group. It's possible to have 3. More than that tends to become unwieldy. And we'd love it if the co-leads are willing to create a Slack channel and monitor it and encourage discourse that way. Although you're welcome to use other communication. We're seeking volunteers and suggestions for additional leads. If we look at the list now, innate adaptive immune response, we have 1 person each. Perhaps they should be grouped into a single area. Generic host pathogen interactions, we have a number of volunteers. You'll see some overlap of names. Tissue damage and recovery, Yi Zhang agreed to do that. We don't have anybody talking about viral transport yet. Viral modes of entry and barrier function, which of course we just heard to talk about. We have a volunteer. We have volunteers for viral replication, volunteers for virus release. In the area of therapeutics and medicine, we have a number of suggestions, but in the area of diagnosis and prognosis, remember that this group is not a COVID group per se. It really is a group trying to look at viral infection more generally. COVID is welcome, but if you're working at influenza, Nipah virus, or something else, that's fine too. So don't feel that you should be restricting yourself to COVID. In the area of individual organ systems, we had a lot of people who were interested in kidney and liver, enough that perhaps we should split those two organ systems. But nobody spoke up for lungs, heart, or nervous system. So maybe we need to do some combining. So we need to think about that. Somewhat surprisingly, maybe because we don't have that many clinical people, nobody bid for the comorbidities, which I think are actually a very important topic. We have some nice, Santiago volunteered to lead some discussions on data side of things. That would be great. AI-based processing, we have some volunteers. And some ideas about people interested in zoonotics, which of course are very important for COVID, but also for other viruses. And then at least one person is interested in emerging and re-emerging diseases. That's what I had, modeling technologies. We had two people who were interested in transport, very important problem, which comes back to what Jacob was asking about, how does the virus get to the surface of the cell in the first place. We had some good volunteers for integration between scales, reproducibility. Bobby Maimanchi is willing to do some work on communication and community building. Reinhardt and Paul Macklin have agreed to talk about crowdsourcing software. And John Rice is willing to lead a discussion group on outreach and dissemination. So that's what we have. So again, you'll see some people's names appear many times. Those people we'd like to try to focus. There are plenty of subjects which don't have topics. We'll give people more or less until Wednesday of next week to self-organize. And I'd like to turn it over now to Reinhardt to run a discussion about what to do now. Let's talk about this for a few minutes and see what people's ideas are.
But you're welcome to continue. You've been doing great. So in any case, what we've been discussing is, what should the charge for the individual subgroups be? In some sense, it's not so different from the charge for the entire group, which is in the proposal that we submitted to the MSM steering committee. I think the very first issue is really to sort of take stock in what there is, and we just saw this in the discussion following Yaling's talk, is what are the key issues, what models are available, what data are available, and what is still missing. I think that in itself needs to be step one, I think, in sort of reporting out from this group, as well as catalyzing research and directing it to the places, I think, that are most in need of attention. That's really as far as my ideas go, our ideas go. There are some bullet points here, but like all of this, it's a community effort, so we would very much like to get input from the community as to what needs to be done, and they might very well be different for different subgroups.
So if I might mention something that seems peculiarly missing is the word epidemiology, right? I mean, at least when I come to viral pandemics, I'm thinking about transmission, and there's mention of interspecific transmission, but we have nothing on within human transmission, which is a multi-scale problem since it happens in rooms, in states, people get on airplanes and so forth. Is that excluded from this for a reason, or is that something we should include?
So nothing is excluded per se, but because epidemiological modeling was so advanced, and there are so many forums for the discussion of epidemiological modeling, the main focus of this working group is on in-host modeling. in-host modeling and its connection to epidemiological modeling definitely is something we want to understand. In other words, if there are things that we could use in-host modeling to do that would be useful for epidemiological modeling, that's very important. Reducing susceptibility, reducing the degree to which you spread virus and infectivity, these are very much things that could be addressed. And certainly we want to hear about epidemiology, especially in the context of what you could do if you had more detailed single individual models. But the goal of this group was not to exclude epidemiology, but rather to provide a forum for the areas that were less discussed, because their MIDAS and other programs are so extensive, focusing on the epidemiological scales. The reason there was a specific discussion of zoonotics and animal human transmission was because it's a somewhat understudied area. In particular, in-host dynamics, for example, of COVID in bats is a very interesting problem because bat immune response is so different from human immune response. And you could ask some very interesting questions about, for example, does bat immune response lead to pre-adaptation of viruses that might be significantly damaging to humans because of those differences. And so that was the reason I think that particular item was there. We also had people who were speaking up for it. We certainly don't want to exclude epidemiological modeling if there's strong interest. But again, the focus here is balance.
Yeah, just to follow up on the discussion at the end of the talk, thank you for the talk, by the way. To me, sort of the rubber hits the road with dose response, right? How many, is what we're talking about. How many viruses do you breathe up? Where do they go? How sick do you get? What's the probability of infection? To me, that's a critical issue. And I was investigating a little bit for my class, and we don't know much. I think we could have a huge impact in trying to integrate things through the dose response.
Fred, for COVID, contact me. I have a bunch of literature I can send you. But specifically to what James said, what happens within the body, let's say treatment by some sort of medicine or anything, we still don't know the effects of many of them. So you cannot even model them. I encountered this with diabetes a long time ago, but now it's also like with COVID. They'll start vaccinating people. How effective is the vaccine? This is one idea. But we need this information. And yes, I agree with you that the information outside about how people transport, how they move around is important. But this information is also important, and some of it is available in the literature, some of it is not. I can point you things that are missing today still.
So Fred, I completely agree with you. I think it's an excellent point. And depending on, the scope of this problem, that might well be the topic of a specific subgroup that focuses exactly on that issue.
I think that the issue of dose response is very interesting. If you have a higher initial exposure, do you have a more or less severe progression of the disease? There's some data sets in some diseases where you have very paradoxical effects, where if you have low initial exposure, you don't mount a strong interferon response initially, and you wind up having a worse progression of the disease than some higher exposures. Usually, if it's very high, it's bad, but there could be nonlinearities and non-monotonicities that are very interesting. So I think those are great topics. Again, the goal isn't to exclude anything, and bringing up these issues and what is the missing information, I think, is critical.
Perhaps what we should do is generate a subgroup that specifically to liaison with MIDAS and other epidemiological groups. Not so much to duplicate their efforts, but just so that this working group knows what, for instance, MIDAS is doing and MIDAS knows what this group is doing. I think that would be a great topic for a subgroup.
John Rice. Because I also work with the pandemic prediction forecast group, and I really am looking forward to talking to Fred. the relationship between the kinds of things that the biological computational biologists are doing and the social aspects of the epidemic modeling, where the models that we've seen being used they don't have any biological data about the virus itself when they try to determine what the probability of transfer, if people come in contact, how long do you have to be in contact? You cannot answer those questions without a lot of detailed knowledge of the virus organism itself. And that the real opportunity here is to focus on what is it that the prediction models need to know first. When that brand new novel virus shows up, what are the most important questions that we could give creditable input to, instead of making them take wild guesses for their assumptions? And so, although the group may not get into the epidemic prediction model itself, the output of what we're trying to do is the first most essential input to the prediction model. You can't do it until you understand the organism.
Okay, Reinhardt, I think these are very good points. I don't know, why don't we go through the last administrative slide? We just have one or two, and then in the remaining time, because we're going to run out of time in 7 minutes, people can stay on if they want to keep talking, but I don't want people to have to stay on.
Yeah, I don't know that there's much at this point we need to get back to. I think it's a great example of how this group self-organizes around scientific issues that come up. And you know, if people are interested enough, they will take initiative. So in this case, I could see that Fred and Jacob would just be perfect co-leads of this subgroup, and probably would get lots of people to join. So I would suggest that we'll just do that. And as we go on with these discussions, I think there'll be a number of other opportunities where things like that emerge.
I certainly don't want to cut the discussion off. I mean, the only other things, please look at the website, review contents. It was thrown up in a hurry. We can change the charge and the text. For next week, suggest weekly discussion topics. This issue of connection, and for example, just the issue of infectivity and progression of the disease as a function of initial exposure, that could be a great topic for a Slack discussion for a week. If people have suggestions for items they'd like to see in the agenda for next week, please let us know. Suggestions for mini speakers. If people are willing to take the time to send us a one-pager on your research and interest, we can post those on the website to have a little bit more of an introduction. Last time we went around the room and everybody introduced themselves in 1 minute. If you'd like to introduce yourself a little more, that's an opportunity. We would very much like to have a preliminary list of subgroups and leads by the middle of next week so that we can begin to sign up membership for subgroups. We created a Google Doc, which was just a placeholder for people putting in discussion items. Nobody edited it. I don't know whether that's because you couldn't get to it or because nobody was interested.
You should publish it once more. I didn't see this.
So Jim Sluka, could you paste that link into the Google chat, please?
Which link? The Google
The link
Doc?
to the Google Doc. Okay. There are a couple of links you'll need. The spreadsheet of subgroup leaders, where people should indicate their preferences of subgroup suggestions for merging or splitting subgroups and so on. And then we have essentially just a Google Doc, which is an empty space where people can put ideas and comments. You could use the Slack as well, of course, for this. And so I think that was really the end of the main meeting. If people have ideas for people to invite, please feel free to send that invitation around to your friends or to people you think are important to include. Again, people are welcome to post tools and software to the IMAG MSM website as long as you have appropriate disclaimers on it. Once again, we had our own registration form for the working group, but if you want to post directly to the IMAG MSM website, you need to register separately for that. And so to do that, you have to go through their registration forms. It's not the most trivial thing to do. We'll post this slide deck online, and the instructions for how to do their registration are available in the slide deck. I'm not going to take the time to do that now. I think that was the end of the formal business of the meeting. Reinhard, is there anything else you want to talk about?
No, I think that's all. Thank you.
If anybody has any other comments or questions that you'd like to address now, we can do that in a few minutes. And I thank everyone who has to, people have to leave promptly at 4, I understand. I thank everybody for showing up. It's always hard to get these things started. I thank Yaling very much for giving us such a stimulating talk on a scale that we don't, many of us think at different scales. And so starting at the molecular scale is a great thing to do. So thanks everyone. And if you want to talk more on an open forum now, you're welcome.
Thank you, James.
Thanks.
Are we still recording?
Yes, I can stop the recording if people would prefer.
If it's open talk, may as well stop.
Are there multiple people who would like to stay on for discussion? I don't know. Let's see how many people would. Jacob would. I don't know how many other people are available. So let's see. Maybe it may just be Jacob, you, me, and a few. In which case,
James, Bob Stratford here.
yes.
I just wanted to say one thing to add to Yaling's very good talk. So, you know, he's talking about developing, you know, compounds that inhibit the ACE, you know, the binding of the virus to the ACE2 protein. And so, you know, when we're talking about small molecule inhibitors, inhibitors, you know, we need to think too about, you know, how those inhibitors, you know, get from the tongue when a person takes a drug all the way to the site of action, you know. And so we're talking about absorption, we're talking about distribution from the systemic circulation, you know, following absorption to the site of action. And then, you know, in parallel with that distribution to the site of action, there's elimination, which is always going on and always reducing drug concentrations. So, you know, that's another very important aspect to think about, in addition to modeling, you know, the larger scale, you know, the whole infection process, which I felt that we were, you know, we were talking about that, but I just wanted to add to that, you know, when we are talking about a drug that's going to inhibit a target or alter, you know, a target's expression, that we need to think about, you know, how that compound gets to the site of action and all the processes that go on to influence its concentration over time at that target. It's a very complicated process, and I guess it speaks to that drug development subgroup, which is a real big, you know, subgroup. There's all kinds of stuff that goes on in drug development. So anyway,
I think those concerns about drug availability and dosing, and the PBPK downstream of that, are really critical, and I hope you'll represent that set of ideas, maybe be able to recruit some of the pharma people to participate, because I think so far we don't have that many biotech type drug people on the panel, and it would be wonderful if we had some representatives from some of the main pharma who could address what kinds of questions they'd like us to solve.
Yeah, excellent point. I had the same thinking that I noticed last week that I didn't see any pharmaceutical industry or biotech industry representation. So that would be a good thing to do, would be to try to recruit people.
If
from
you
those
have
centers.
connections at Lilly, Pfizer, or any of these, Gilead, any of these companies,
Yeah.
there's so many different communities, and the overlap is not zero, but it's not large. And so one person reaching out and crossing those barriers is really valuable to us.
Okay. Yeah,
It would certainly be really useful to start off with just a liver-centric PBPK with representations of, for example, dexamethasone. As a kind of right now, basically a gold standard for a treatment, and whether it's good or not is somewhat irrelevant. It's what's being used, and there's certainly a massive literature on the ADME of dexamethasone in humans.
Sure.
And that would kind of give, at least put a stake in the sand that we can pivot around a little bit.
Good idea.
So
Yeah, I'll
certainly
look
an
into that.
ADME group. Okay, that's good. An ADME group would be a good idea, I think.
Great.
So please feel free to add that to that list of subgroup topics. And again, you don't need permission to send that invitation letter out to people.
Okay.
And if there are forums, if there are forums in the PBPK or pharma community where you could post that invitation, that would be fine. We'd very much appreciate your help with outreach.
I can give you an example of what was done in the past within diabetes, for example. I was working on diabetes model. One of the things that I was at that point slightly involved with is looking at how different medication affect diabetics at different stages. And they do affect what other parameters of the person. For example, some of those drugs lower the weight, and some of the drugs, once you start taking them, they make the weight higher, which changes the risk profile of the person to have a disease and things like this over long periods of time. So you can see like diabetics that are pre-diabetics will take certain type of drugs that make their physical properties differently, and then they will start getting insulin, and their weight will rise up, and those will affect some other parameters. And think about it, all those clinical trials that are being run, you can actually extract some information from clinicaltrials.gov exactly about what drugs are this group taking, and then you can add this into a population model, which actually I'm doing already. So if you can actually come up with, some of the team here is interested in diabetes, for example, I have the other transport to take this information and plug it into an existing model, actually multiple models. But this is, and then you can play around with this and figure out which one of those assumptions, how effective or ineffective they are, about really how much the drug is effective or not in the long term.
We're going to the park.
I think this issue of diabetes, I was a little disappointed nobody picked up any of the comorbidity subgroups.
It's a viral infection. Virus is in the name of the working group. So it's like, sure, I'll do diabetes. I've been doing diabetes forever. But the thing is, I'm more interested specifically in integration. I was looking, many people are interested in disintegration. This is why, I'm trying to see how we can integrate things.
Well, I think what we need is at least one or two people to be a leader for any of these sub areas. That doesn't, being a leader of one subgroup certainly doesn't mean you can't contribute and participate very intensively in others.
Sure, but put me under diabetes. I have cardiovascular diabetes easily, so you can put me under if you want to. I'm more interested, however, in the integration, because this is really what it's all about. But that's me.
Well, it's. I mean, I think these are all important problems. It's just simply that, as a practical matter, if we don't have a leader on on the area, it's unlikely that much will happen, because it requires a lot of work to herd the cats, as you see here in the video, to get something to happen. So I think Reinhard had to leave because he had another meeting. So I think probably we should bring this meeting to a close now. It's great, Jacob. Thank you very much for your contribution. And I look forward to having you being an important part of this group.
Sure.
And Robert, if you can reach out to the pharma and the PBPK worlds that would be fantastic. We really would appreciate that. Okay, well, thank you very much, everyone. Now, John, thank you. You got your discussion of zoonotics, which is one of his passions. So thank you, everybody, for participating. I hope it was interesting. And next week we'll have 2 talks, and Gary Ann is always a great speaker. And the fact that Alan Perlson's willing to talk is, I hope you'll remember, because he's a busy man. It was great that he was willing to be to address us. So, and Jacob, we'll look forward to having you schedule your talk soon.